Start recording.
Okay, so should I start or do I just waiting
Yeah,
for
yeah.
some?
You can start. The others can catch up.
Yeah.
Yeah.
Okay, let me just move. Okay, so today we'll be getting into the nitty-gritty of what quantum biology is and what is not. And we have in the previous two lectures had a lot of background information about quantum physics and related fields, quantum chemistry, as well as quantum statistics. And now we've reached a point where we can try and understand how if there are any existing examples of quantum effects in biology, and whether we can make a connection between these two gentlemen, Schrödinger and Darwin. And so one of the arguments in favor of quantum biology is that the human civilization, after about 10,000 years of its technological progress, at least, has discovered quantum physics as the main, the most fundamental theory of how matter interacts with itself, and and that has led to tremendous technological progress, as I tried to elaborate a bit last time. So it would be hard to imagine that the evolution on this planet, biological evolution, as explained by Charles Darwin in terms of survival of the fittest, adaptation, improvements, and competition between species on the basis of fitness and survival under various conditions, would not have achieved the implementation of quantum effects because they are so powerful and so advantageous in many situations. So two billion plus years of evolution, not only that, but parallel experimentation. So basically every new um, organism born on this planet is a unique experiment in terms of survival and competition with other organisms, and of course species on a statistical level. So that is an argument um, for in favor of quantum biology on the basis of evolutionary retention of adaptations and improvements. So yes, that's what it says here. And as I mentioned, Schrödinger in 1944 wrote this book, What is Life? And with, of course, he was one of the main architects of quantum physics, but also thought about biological systems and suspected or proposed the presence of quantum effects in biology. Um, so at the same time, we have, of course, achieved various implementations, but there are still technical issues with advances in quantum technology, including quantum computers. As you probably know, it's now after more than 2 decades of development, there are several quantum computers operating with great efficiency and competing and outcompeting classical computing. I think very recently there was an example in talked about in the media about quantum supremacy being achieved by some quantum computers in terms of speed and performance. So, all right, that's great. If quantum computers outperform classical computers, then probably this is something that Mother Nature has been able to discover as well. But there are these persistent criticisms and doubts expressed about whether it's actually possible. And they stem from the difficulties when we develop quantum devices to keep them operating at peak level, and one of which is environmental decoherence. In other words, interactions with the surroundings that destroy things like quantum coherence. So that means well composed superposition of states. Then, of course, that leads to attempts to isolate these systems thermally and physically, and maintain them at low temperatures, because temperature, by and large, is the quote unquote enemy of quantum states. Yes, that's that. And warm, and then lastly, wet biology. You know, most of the biological systems are composed of something like 70% water. Fluid state is not really where quantum effects have been seen. Usually it's solid, due to less dynamical mobility of structures and more organized architecture, symmetrical, etc., etc. So these arguments have been raised, but there are always ways around them. And so this is the maybe incomplete list of arguments against. And this is not a very good argument that cells are big. Well, I gave you examples of microscopic quantum states and systems such as magnets and crystals and so on. Hot, maybe an exaggeration. Biology is not hot, but it's warm. At physiological temperatures of over 300 Kelvin. So in comparison to things like superconductivity at 100 and maybe 20 Kelvin now, I don't know exactly, but around there. That's really quite a bit higher, but at the same time, we have lasers operating at basically physiological temperatures, room temperatures, and higher. These are quantum devices. The difference, of course, is which we'll discuss today, and kind of demolishes some of these arguments is power supply. Okay, the quantum systems, with the exception, of course, of lasers and quantum computers, are at equilibrium, but lasers as well as quantum computers require a steady supply of sufficient, above a certain critical limit, external power. And some people who criticize these things, forget it. In biological systems, of course, power supply, not supply, power supply. In biological systems, this is sine qua non. There is really no life without continuous provision of nutrients, which then are converted into metabolic energy, and so on. We'll come back to it. That may be a bit of a problem, which I'm not going to discuss, but just to mention that there are some very serious studies showing that water around biological structures such as proteins and DNA is in a different state. Jerry Pollack wrote a book, The Fourth State of Water, which is between liquid and solid. Let's say something like liquid crystal, ordered water, fourth state of water. Jerry Pollack is an eminent professor of biophysics at the University of Washington in Seattle, and he has produced some amazing work, which the audience here can find on YouTube as well. There are excellent lectures given by Professor Pollack that explain this phenomenon, and I think that may be the key to dealing with this argument about wetness. Now, life is slow. Well, we talked about time scales. This is a very, I would say, simplistic argument, because life is maybe not slow. It depends on which process you look at. There's a spectrum of frequencies and time scales from atomic to geological. So we can't say that. What we can say is that the mystery is how different time scales overlap, and how scales of spatial dimensions and scales of temporal dimensions inform each other, and probably both top-down and bottom-up in a hierarchical manner. And that to me is the real question, rather than saying slow or fast. It's I would say life living systems have processes which are both fast and slow. That's more correct. Complexity, I think complexity is only in the eye of the beholder. For the purpose of modeling, it makes our modeling difficult. It doesn't mean that complex systems cannot be quantum. That's, again, is a very, I would say, silly argument, especially when you look at, for example, superconductors that initially were quite simple. And then ceramic superconductors, actually with structures which are much more complex than early superconducting systems, became superconducting at higher temperatures. So complexity is, again, another good argument. The fuzziness, again, that's a misnomer. Depends on what we are asking. Whether biology is real, well, it's, again, in the eye of the beholder. Whether it's fuzzy or real, we need to look in more detail. There are arguments about cells being in quiescent states between living and non-living, viruses being alive and dead, depending on the conditions. If there's a host, they will start being alive. When they are in an environment without a host, they will be dormant. So I don't know. That's, again, not a very convincing argument. So realism, again, is our model of reality. Whether we believe in realism or in determinism or in Heisenberg's uncertainty, well, that's not a question of belief. It is a fact. But whether this extends to biological systems is really subject to our own interpretation. Okay, so all these arguments, in my opinion, are weak. The only truly challenging one is this: how hierarchies, just like Russian Matryoshka dolls, interlock and form a coherent whole. How you can organize and synergize pieces of that big puzzle, even at the level of a single cell, from atoms to molecules to biomolecules, macromolecules, assemblies, and eventually an entire cell. That is, to me, the most interesting question. Okay, now this is a list of features which sort of molecular biology and biochemistry, structural biology don't really know how to address, in my humble opinion. So how to do collective dynamics with many degrees of freedom in systems out of thermodynamic equilibrium, which is clearly the case with living cells, which are in a metastable high energy state compared to the stable thermodynamically equilibrium state that is the state of death. Then how do you combine local and global order? And this is what I just talked about, hierarchy, dynamical hierarchy of temporal and spatial scales. Complexity, again, that's really, I would say, a problem that is technical, maybe not a deep philosophical problem. Short and long-range correlations. Long-range correlation in particular, there's really nothing in the arsenal of any of those branches of biology that can explain long-range correlations, communication across large distances, connecting different parts of the cell and beyond. And as it states here, for physics, really the challenge here is that these are open, irreversible, and dissipative systems. I would add to it highly nonlinear. Nonlinearity is a property which states that the cause and effect are not proportional to each other, and that's clearly the case with biological systems. So self-organization, there are some explanations of this from the nonlinear dynamical systems theory. It doesn't have to be quantum, but nonetheless sophisticated approach, mathematical approach, which goes back to Ilya Prigogine and Hermann Haken, Haken's synergy, Ilya Prigogine's self-sustained catalytic reactions. Sorry, I mentioned here nonlinear interactions, yes, and highly integrated dynamics. So that these are the features which, some of which at least, can be either explained by very high-level nonlinear dynamical systems, descriptions in mathematics, or by the use of quantum physics or quantum field theory, etc. So now this is what quantum biology typically refers to. My personal definition is broader than this, but here's what Wikipedia states. So this refers to applications of quantum mechanics to biological objects and problems which are non-trivial, acknowledging that trivial applications are really quantum chemistry, because all chemical bonds pretty much are quantum in nature. Therefore, this level of organization of matter is quantum by definition. But whether it propagates beyond to higher levels is open to discussion, and quantum biology says yes, it can be seen at a higher level. And so non-trivial here means all these characteristics of quantum systems which are simply not present in classical physics. So superposition of states, no single value. We are trained or conditioned to think in terms of deterministic, strictly deterministic understanding of material universe. So, but quantum mechanics dealt a blow to it, and a quantum system can be in several states at the same time, and only when you perform a measurement, it has to respond with a so-called collapse of the wave function and manifest a given state. But that's the the metaphor for it is, of course, the Schrödinger cat that everybody hopefully knows about. This thought experiment, rather cruel, but a Schrödinger cat, but illustrates the seeming absurdity of quantum mechanics, but in fact, it's the true nature of reality. All right. Now, non-locality, this is a very big departure from classical physics, which means that there are some non-local interactions outside of the four forces of physics that lead to things like entanglement um, um, and the arbitrarily distant systems can in some way communicate to each other the state in which they are. Right, so entanglement and tunneling is another bizarre phenomenon, which obviously is well tested experimentally and applied technologically in things like semiconductors. And that means that quantum particles with energies which classical physics would preclude from enabling some transitions, they can overcome potential barriers and tunnel into another place, location, by a little bit like throwing a tennis ball against a wall, and in the classical world, this tennis ball would bounce back to you, but in a quantum world, part of it would go through it, and part of it would come back to you. So it's really strange, but uh, correct without a shadow of the doubt. So then there's the Heisenberg uncertainty principle, which tells us that we cannot determine the actual state of the system with arbitrary precision, especially to conjugate variables like position and momentum. So these are the most bizarre quantum facts and non-trivial features. So quantum biology aims to find such non-trivial features in living systems. And question is, did quantum biology find it? And the answer is well, yes, and yeah, and no to some degree, but largely yes, but not many. And this is a list of luminaries in science of the previous century who advocated for the essential role of quantum mechanics for our understanding of life. And so we have the famous quantum mechanics pioneers, Niels Bohr, Werner Heisenberg, Erwin Schrödinger, John von Neumann, Carl von Weizsäcker, Elsasser, Weizsäcker, Eugene Wigner, Freeman Dyson, and Arthur Kessler, and others. So now we can ask, are there any, let's say, reflections of quantum behavior in living systems? And this is kind of a list that we'll go through in some detail, but this is quantum metabolism is a big one because, as I told you, power generation and transduction, use of metabolic machinery to propel living systems forward and maintain living functions is essential, and it can be demonstrated to be quantum, both in terms of plants and animals. So photosynthesis in plants and some bacteria and algae can be seen as a metabolic activity, because really light, electromagnetic synthesis, electromagnetic radiation, quanta of electromagnetic radiation, namely photons, are absorbed. Their energy transduced into electronic states, which then can be used for this synthesis of glucose. And in animals, we have oxidative phosphorylation in mitochondria. I'll talk about this later today. That is at least has some features of quantum behavior, both in terms of individual states, electron transfer chain in particular, as well as statistical properties. So this is interesting. This is interesting, both super important, and both are essential for life, both for plants and animals. Then there is the well-known phenomenon of ultra-weak photon emission from living systems as a byproduct of metabolism, kind of flip side of generation of energy. This is emission of energy, biophotons, and that has led actually to some pioneering technological implementations in photobiomodulation, PBM, photobiomodulation, which is basically the use of specific frequencies of electromagnetic radiation to treat conditions, syndromes, diseases, including some neurological disorders, etc. So there's something here. Maybe in the last lecture I have time, I can talk a little bit about some companies that develop devices. Sorry, the next one I already mentioned, so it's really part of also a separate issue, but part of the metabolism in plants and bacteria. This was a very highly discussed and published theory of transmission of energy in biological systems at the subcellular level through so-called Davidov solitons. Davidov was a Ukrainian Soviet physicist of great repute, and he developed a theory of how excitons and phonons in alpha helical chains can transmit energy over large distances without loss. So this is a nonlinear quantum phenomenon, in fact, very interesting. And then somewhat related, and about the same time, now almost, well, maybe even more than 40 years ago, Herbert Froelich, another quantum physicist of great repute, proposed an explanation of biological coherence, so coordination, coherence, communication between cells over large distances. Large distances means here on the order of cellular dimensions, so microns. For us, it's short distances, but for molecular biology and biochemistry, long distances, as opposed to, let's say, nanometers. That'll be micrometers, which is 1,000 times longer. So there are some continued efforts to demonstrate this effect, and I'd say some published evidence for this. How widespread it is, I don't know, and maybe not important. Just to show that it exists is enough. And then there are some more speculative theories of quantum electrodynamic coherence in cellular water, developed by these three Italian scientists, Giuseppe Vitello, Preparata, and Del Giudice. So now we can say, having said all of this, is quantum mechanics relevant to biology? Well, at small enough scale, definitely. We talked about quantum chemistry, but also at a slightly larger scale. And here are some examples. Now we're going back to 2000, the first decade of the third millennium. So it's now 18 years ago or so, 15 years ago, where this was a big story, demonstration of the fact that photosynthesis is a quantum process, capture of quanta of photons, electromagnetic waves, by photosynthetic systems called FMO in chloroplasts of leaves, for example, but not only in bacteria and algae, was demonstrated to be quantum. So that was actually the first major demonstrated step toward quantum biology. Another one was maybe not as big, but I would say even more puzzling before the publication of these papers. European robins are migratory birds that fly around this time, maybe a bit later, from Northern Europe to Southern Europe and Africa, North Africa, to spend the winter there. And they have something like a compass built in, so they can find direction and they never fail. How is it? And that was an explanation proposed in these papers on the basis of quantum transitions in the specific molecules in the retina of the eyes of these birds. As of course, a similar phenomenon exists in North America with Canada geese traveling to Mexico and I believe California, perhaps, to spend the winter in a more accommodating climate. So that's that, and this is actually from a review paper about this migratory bird molecular compass. So this is the bird. This is the eye of the bird magnified, and inside the retina that there's a cryptochrome complex, which has it's a little bit technical, but it has electronic structure in the valence electrons. There are two with spin up and spin down. So the ground state is an S state, which means not no net spin, but it can transition under the influence of an external field into a triplet state where you have three substates and they have a net spin, and that spin interacts with the magnetic field, and that causes an electronic transition, which then cascades down to an electromechanical transformation, and that leads to the perception of the change of direction, like a bit similar to a compass needle. The needle moves, and you follow the needle to maintain the direction. If you use it in the wilderness, for example, and the bird has its own internal compass, which informs through a signal in the brain that the direction has been altered, and then that has to be corrected. How that specifically translates into the bird's the brain perception of direction and so on, it's probably a bit more complex, but that's the main fundamental mechanism involved in this. So it's a quantum state. It's so sensitive, it actually is comparable and maybe even beats the sensitivity of some modern magnetic field measurements with current technology. Then there is a host of physiological quantum effects listed here. It's been known for a long time. Of course, the human eye is probably not the most sensitive type of eye among the animal kingdom for detection of photons, light. But even here, I think we have sensitivity to the point that perhaps not a single photon, but several photons can be detected. And I think birds, of course, eagles and hawks have much more sensitive eye. And it's believed that single quanta can trigger electronic transitions in the retina of the eye. So then there's a sense of smell, olfaction, which is, again, humans are not the best at this, but of course, some dogs are, especially trained by the police force to detect contraband or narcotics, and their sense of smell is such that a single molecule in the air can be detected, and there is a theory behind this by Luca Turin and collaborators who demonstrated that this is not lock and key mechanism so common in biology, but it's a resonant interaction between the vibrational modes of the aromatic molecule and the receptor molecules quantum states, also vibrational. So that's a molecular recognition mechanism, which is quantum. Now, yeah, that's very interesting. I mentioned this. So magnetosensing, photosynthesis, I mentioned this. We'll talk about mitochondrial metabolism a little bit more later on. And then there are these more esoteric and interesting but not settled claims about consciousness, how the human brain operates, whether it's classical or quantum. And Roger Penrose, the Nobel Prize winner from 2020, wrote 2 influential books where he claimed that consciousness that cannot at least entirely operate within the classical deterministic framework, and proposed various, well, not various, actually, together with Stuart Hameroff, a theory called OR, which involves a quantum orchestration of quantum states, objective reduction, and their collapse using gravitational forces, and that leads to the formulation of thoughts and things of this nature. So consciousness with a question mark. I'll say something about it in the last lecture on Thursday. Let's leave it at this for now, and that leads also to anesthesia. It's still not completely explained how it works. Anesthesia, as you know, is a mechanism, is a procedure that allows, of course, modern medicine to perform surgeries and all kinds of medical techniques, because it causes reversibly a loss of consciousness of the patient, and then the surgeon can perform the operation without the patient feeling pain. So it's also analgesia here, but mainly loss of consciousness, and therefore, how does it happen? And these gentlemen, Stuart Hameroff and Roger Penrose and others, claim that anesthetic molecules quench quantum states in the brain, and that is the basis of loss of consciousness. Again, I'll try to say something more about this. There are also claims or theories that both the protein folding process, which kind of defies classical calculations, if it's a classical search algorithm in the space of amino acid conformations in a peptide chain, that would take the age of the universe for a single protein to fold. That's called the Levi-Thal paradox. But in fact, it happens over microseconds to seconds maximum. And then gene mutations are also suspected of being not random, but directed in some way, and perhaps even the mechanism of mutation is a quantum tunneling effect. So a lot of things on this on the plate. This is about olfaction. It's basically two competing theories. One would be classical shape matching, which Lukaturin and his collaborators claim has actually no basis. Lukaturin, because the aromatic molecules that are studied here, including molecules of perfumes, really have no similarity in terms of their shapes, but they may have same vibrational frequencies, and that is the characteristic that allows us to sense them. So this is shape matching schematic. As you can see here, shapes must be very specific to match into a receptor, but the vibrational resonance theory says no, it's not about the shape, it's about this frequency of oscillation the molecule executes and is recognized by the receptor. Another interesting thing in terms of biological computation, it could be employing quantum search algorithms, and a bit of a mystery that we have some characteristic numbers, such as the DNA code has an alphabet of size 4. That is, there's four nucleic acids, the same for RNA. The protein synthesis uses an alphabet of 20 letters. Each letter is an amino acid, and things like transcription factors or various protein-protein interactions can be seen as exploring the surface of a DNA molecule or a protein. And interestingly, quantum search algorithms with one query can carry out a simultaneous exploration of a database of size 4, just like in DNA or RNA, and with 3 queries as a database of size 20, which is for proteins and peptides. So there is some, I would say, uncanny similarity. That doesn't mean it's a quantum search. It is rapid, and we know that some of the molecular recognition mechanisms in biology are very fast, and through random walks, diffusion, no explanation can be provided to give the account of this. A quantum search algorithm would. Let me go back to bioelectromagnetic field concepts, which go back to the 70s. And I mentioned these names before, but maybe now a bit more. Interestingly, a lot of scientific research on these topics took place in the Soviet Union. including biophoton discovery by Alexander Gurevich in Russia, actually Soviet Union, and then we have Ilya Prigozhin. I think he was of Russian origin, but lived and worked in Belgium, in Brussels, got a Nobel Prize in chemistry in 1977 for the so-called theory of dissipative structures, which is the generation of patterns and nonlinear chemical kinetic reactions leading to stable oscillators, biochemical oscillators. And then I mentioned Froelich, who introduced the concept of biological coherence. Prigozhin's work did not include quantum effects, but Froelich's did. And yeah, that's worth noting. Froelich theory, maybe I will very briefly explain. He postulated several criteria for the existence of coherent states in biology. One was the sufficient rate of pumping of energy above a critical threshold, just like in the laser action case. The second was presence of thermal noise, which is given, of course, because of physiological temperature. Third was some nonlinear interaction between the degrees of freedom, which led to the emergence of a metastable coherent state, dynamically ordered state, which is this collective dynamic mode, similar to Bose-Einstein condensation. I mentioned Bose-Einstein condensation last week in the second lecture, and so Herbert Froelich claimed that something similar is happening in biological systems, except they require this energy pump. Nonlinearity is, of course, sine qua non. And that was predicted to result in coherent oscillations in this range of frequencies, terahertz, roughly speaking, and that led to long-range interactions and dynamical metastable state. Okay, maybe I don't know if I want to talk about this much. I just want to say that when you look at the cell, beyond the obvious, I mean, this is the structure of the cell. This professor from Northwestern University in Chicago developed a theory and did experiments demonstrating some form of intelligence of the cell and cell-cell communication without the presence of chemical or mechanical interactions. And that's kind of interesting in connection with quantum mechanics, because that could involve various basic quantum states at some level. So let's say the following: the membrane is the site of proposed coherence by Froelich. Membrane dipoles are, according to Froelich, oscillating with a certain frequency. Then we have actin filaments in the cortex, so-called microfilaments, and in the cytoplasm we have something which we'll come to appreciate shortly: mitochondria, energy production, organelles, intermediate filaments providing shape and structure, and microtubules. Here we have the center of cell intelligence, according to Günther Albert Bühler, namely the centrosome with centrioles composed of intricate structure of microtubules and emanating microtubules, which are instrumental in cell division, as well as a lot of processes in the human brain in neurons. So we'll come back to microtubules on Thursday, but here we're not talking about cell division or neuronal processes. We're just talking about cell-cell communication. So he published this in very reputable journals, including Proceedings of the National Academy of Sciences of the USA, and he claimed that the so-called eyes of the cell, maybe an information processing center more than the eyes, are the centrioles, and they are able to detect near-infrared signals. He determined the sensitivity to these ranges of wavelength, very close to the red. The visible range of wavelengths in the electromagnetic spectrum is from 400 to 750 nanometers, and his experiments, as you can see, the peak is certainly in the infrared. It's about maybe 1,000 nanometers, 950 or so, but there's some sensitivity still at that would be 600 here, so that's visible. Up to here is visible, and not only that, not only sensitivity to light, which is now shown by this experiment with infrared light source trying to mimic another cell's presence, but also processing and executing movement toward the source. So these are the centrioles that are supposed to be the centers for information data processing from the external environment, and they are composed of 9 triplets of microtubules shown here, with a central cell, with an axoneme, in the center. So it's a 9-fold symmetry, very symmetrical and very well organized. This topic, I need to move on, but this topic is very interesting, and that's the way things work, beyond touching mechanically and sending chemical signals. There's also evidence for cell-cell communication at a distance, which could be due to electromagnetic communication, sort of riding on Albert Buehler's ideas in the so-called bystander effect. And there were some experiments, many experiments done on cell cultures where only, let's say, ionizing radiation targeted one cell out of thousands, and that the cell responds with some, let's say, repair mechanism or overexpression of heat shock proteins, HSP90, or things of this nature. And it was discovered that cells which were not targeted, even quite distant from the targeted cell, also responded to this danger signal. And yeah, this is interesting. And that includes not only radiation, but also chemotherapy, chemical damage to the cell. So there's a stress-induced bystander effect that has been measured, and it goes beyond cells and cell cultures, but also to organisms or tissues, maybe in organisms. And I want to draw your attention to the work of Professor Carmen Mothersville from McMaster University in Hamilton. She published some intriguing papers demonstrating these effects taking place between organisms. She published some work on, and cited here, on, for example, tanks with fish and fish were stressed in one tank, and the next tank there was the same species of fish which were not stressed out by radiation, and both responded the same way. So there is something very peculiar, but potentially explainable by electromagnetic communication, and that could also be in the form of photons and photon reception and transduction of these signals to actionable behavior. Okay, so I mentioned to you that all life needs energy, sufficient energy supply, and so light for plants, nutrients for animals. I mean, plants also need, of course, some nutrients, microelements, and water, and so on. But basically, this is the division that we want to pursue, and the output of it is the formation of high energy molecules, ATP or GTP, mostly ATP, GTP to a lesser degree. And so bioenergetics, by definition of the fact that these things are, these molecules are produced in integer numbers, so not 1, 2, but let's say a trillion, not in fractions of it. You have to have quanta, so we call it biological energy or biochemical energy quanta. This is quantum metabolism, and the neglected player in this game is the mitochondrion, where ATP production from ADP, from glucose really, takes place, but glucose is used to transform ADP into ATP. So how do we know that this is quantum? Actually, it's a long history of studying metabolism in different species across the animal kingdom, actually going back to some very famous scientists, Laplace and Lavoisier, and then later on Rubner and Kleiber in the 1940s. And what all these people found was something interesting, that there is a scaling law. It's called the allometric relation between body size, which is W here, and metabolic energy production, metabolic rate. There are different names for different measures of metabolism. It could be basal metabolic rate, field metabolic rate for animals, and also maximal, but we don't need to go into details. What is important here is that this coefficient beta, so it's W to the power beta, is almost never 1. It is a fractional coefficient, less than 1, so it's sublinear relation. In other words, what it means is that you increase the size of the animal by a factor of 2, the energy requirements increase not by a factor of 2, by less than that. And in most of the cases studied, especially unicellular organisms and animals, have a coefficient of beta exponent 3/4 and sometimes 2/3. Very interesting fractions. Seldom 1. So these are things to remember. It's how shall I put it? It's the economy of scales. So the bigger it is, the more efficient things are, maybe organized. You can think about this this way, but there's also something deeper. And this was that's another slide about the same thing. And this was studied and published by Professor Lloyd Demetrius, a well-known mathematical biologist, Demetrius, mathematician actually, who did a lot of amazing work in biology, and I had the pleasure of collaborating with him. So he pointed to the metabolic enzymes at different levels. You have bacterial membranes with metabolic enzymes. You have chloroplasts with that utilize energy transduction from photons, and then you have the mitochondria, where you have a complex, very complex situation with in their mitochondrial membrane containing 5 membrane proteins complexes, 1, 2, 3, 4, 4, and 5 is the ATPase, FATPase, which perform very interesting, intricate dance of protons being pumped across these membranes, and here you have the number of protons, and simultaneously driving electron transfer. And this electron transfer is a quantum phenomenon, because there's tunneling of electrons from complex to complex and inside the complex over distances, which cannot be traversed and potential landscape by classical means. So this is quantum tunneling par excellence. So we have quantum electronic transitions, and you also have quantization of the output in terms of ATP molecules coming out to these FATPases. And okay, so that's the mechanistic explanation, but how do you explain the allosteric laws of physiology? And this is a bit, maybe I'll skip a little bit because it's a bit intricate. And now you see a bit more in detail the electron transport chain in the oxidative phosphorylation system in the mitochondrial membrane. Okay, this is also based on Nobel Prize winning theory of Mitchell, who explained the importance of pH gradient across the membrane that drives this process, because of the proton transfer that we have to have a gradient. Okay, so 3 building blocks of Lloyd Demetrius's explanation of metabolic scaling laws are these. We have the chemiosmotic coupling by Mitchell, 1970, the energy storage in Froelich, but most importantly, the Debye, that's another Nobel Prize winning achievement, the Debye theory of specific heats of solids. I talked to you about this last week, and Debye, let's say, corrected Einstein in this case and assumed that these oscillations of atoms in a crystal are coupled, and that causes waves propagating through the crystal. And that was enough to achieve proper quantization and proper statistics, which led to the specific heat of a solid behaving like temperature to the power 3 at low temperatures. This observation by experiments was explained by Debye. He got a Nobel Prize for this. And like Demetrius, put it all together, and especially the last piece, and explained that in normal, healthy organisms, enzymatic oscillators in in mitochondria across a tissue or organism are, for efficiency purposes, correlated, and they actually do something like this, form oscillation waves. So there's synchronization, and he called it quantum metabolism. The metabolic enzymes work in concert, and therefore the energy production scales with the frequency of these oscillators, and there is something which you can call the analog of the Planck constant, and we'll call it biological Planck constant. And if you do this, there's a bit of math which I don't want to make it tedious for you. Anybody who's interested, you can consult this paper from 2003, allometric scaling laws of organisms and connection to quantum statistics. This is the key. This is the biostatistics explaining. Okay, that's the math that you can omit for now. Anybody who is not comfortable, you can close your eyes. But if you're into the mathematical equations, you can follow it. And if you do this, that's the way the bi-specific heat is typically calculated. But this is now done for oscillating enzymatic reactions. You obtain then these laws for the energy as a function of time, and then you divide it, take the derivative with respect to time, and obtain metabolic rates. So P will be this is the power or metabolic rate, du d tau in this case, and obtain. And then there's an empirical fact that tau scales with the quarter power of size. So you then put it all together, and that's the sort of crowning achievement of the Demetrius's theory, where it shows that metabolic rate P scales with the weight of the system to the power d over d plus 1, d over d plus 1. Let's see what that means. And by the way, there is this exponential factor, which looks like an Arrhenius relation that comes from the Mitchell theory. All right. So if d equals 1, let's call this beta. So beta equals d over d plus 1. If d equals 1, beta is 1/2. Beta is the exponent I told you about, which is not 1, or seldom 1. If d equals 2, beta is 2/3, which is one of those, another common scaling exponent. If d equals 3, beta is 3/4, the most common exponent. So you see, all of the cases which were listed before and observed by Rubner and Kleiber and Lavoisier and others can be explained by dimensionality of the metabolic networks. And that's the story. This is a very busy slide, which explains this biological Planck constant and the relation between the energy and frequency in enzymatic reactions. The bottom line is this, and I don't want to go through it, there's no time, but the Planck constant in physics is, where do we have it? Let's say 10 to the minus 34th joule seconds. Oh yeah, here it is. And if you do the calculation for metabolic enzymes with some known numbers of enzymes and the frequency of enzymatic reactions, you get this value, which is different. So that's the biological, I would say, cellular equivalent of the Planck constant, and has a ratio of about 2 times 10 to the 11th. It's 200 billion times greater than the physical Planck constant, which is tiny. But this corresponds really to the number of atoms per the area in the cell which surrounds a single mitochondrion. So it makes sense because it really refers to larger objects. Okay, I think I'll just wind down in a minute or two. I mentioned photosynthesis. That's really metabolic process in plants, and that goes back to these papers in Nature 2007 demonstrating this is called the FMO, Fenner-Matthews-Olson complex, and this is bacteria in hot springs in New Zealand performing similar energy capture from light, and that's a complicated schematic of how light gets transferred through different parts of that chlorophyll, and eventually ends up in a complex which has a funnel, and that funnel this is the complex of 7 aromatic rings, microcycles, but okay, each of which is shown here, and this is where the energy capture occurs, and this was demonstrated to create a collective quantum state. So all these 7 molecules, quite large in fact, are reaction centers, and they have a collective wave function. So all of them are in a superposition state with each other. That's another representation, and that leads to the delocalization of electronic energy states. So that there were some very intricate experiments performed, very precise, and also theoretical papers explained the results with great precision. So that's also an example of a quantum search algorithm I told you about. This group of authors showed that this is a search algorithm for the quantum state to find the lowest energy. And these single-celled algae in Australia perform this quantum coherence effect as well. And there was, in addition, there were quantum entanglement demonstrations. So these are entangled states, delocalized collective states in the system of molecules. Okay, let me just skip through this. Yeah, let's stop here. And I will in the next lecture on Thursday, I will go back to microtubules and talk about potential quantum effects in the brain. So that's it for today.
So why don't we talk about anesthesia? We've never had surgeons taking the course before, and now we do. So Ryan's livelihood depends in a constant manner on anesthesia working. If that stopped happening, he couldn't practice.
Yeah.
So, Ryan, in your training thus far, how does what you thought was the mechanism of anesthesia jibe with what you heard today? Are they completely different or some?
To be honest, our exposure to anesthesia is, I mean, quite limited on a formal level. So obviously we interact with anesthesia every day, but our rotations through are really only 1 month long,
Huh,
and the
okay.
deep dives into the mechanisms are not something we go through. Our focus is on those rotations is kind of how do you induce the patients? How does this change them? The physiologic
Yeah.
parameters that we work with, and so this is nice to see some of those underlying mechanisms on a deeper level that maybe we don't discuss otherwise.
Yeah.
And if I may, we'll come back to it. I'll say more about it because today was just very superficial. Because next lecture will be about neuroscience, and of course that fits into the neuroscience bit. And I will spend more time on a potential explanation of anesthesia. Yes. So there'll be a few minutes of deep dive into what anesthetic molecules do, and what this may mean for quantum biology. I just want to say that the main conundrum here is that anesthetic molecules come in various, just like with olfaction for organic molecules. They have different shapes and different sizes. Even a single atom, xenon, is an anesthetic, and there is really no basis for a receptor-based explanation, and of course it goes deeper. I don't want to oversimplify it, but it's a very important topic. 150 plus years, I think, of the use of anesthesia, successful and incredibly powerful tool, and yet we are still we don't have a complete understanding of it. Some of it, yes, and from the technical, as a surgeon, I'm sure you're very confident that the anesthesiologist performing the procedure knows how to do it, and it's 99.99% safe, and and works like a charm. That doesn't mean that we know everything about it, in spite of the fact that it works perfectly.
Absolutely, yeah, I totally agree. And even in having had brief discussions before, I think that even what most anesthesiologists would say is, we have an idea of what these do, but we don't actually know how they work inside of the brain. Like, how is it inducing anesthesia and amnesia and those sorts of things? There's still lots of it that's unknown, even amongst experts.
Yes, yes, and by the way, I wrote some papers on the mechanism, and collaborated with anesthesiologists who are equally puzzled. And anyway, it's a great tool. Let's leave it at this. But if it's a quantum, this is seen as the best reversible, safe insight into quantum consciousness, if we can call it this, or quantum mechanism behind consciousness, right? Nothing else is as clean cut as this, off and on.
Great. Okay. Well, thank you. And another great lecture. And yeah, we look forward to Thursday. And thank you
The
very
grand
much
finale.
for today. Yeah. So, you can think about the future of the course. But it would be nice, we have two cardiac surgeons now. It would be nice if someday we had even more than that. I think that would be uber cool if that happened.
And I would like to hold you to your word, the promise, I guess, to help me record, high quality lectures, many more actually than these four a
Sure.
series.
Yeah. Yeah, no, that's fine. You've been part of a natural experiment of audience retention, in the very beginning, when you said all the textbooks will be rewritten, and
They
thousands
will
of
be.
people came back
I don't know when, but they will be.
yeah, yeah, yeah. Okay,
Uh,
we'll see you on Thursday. Thank you very much
uh,
for today. Okay,
Thank you. Thank you all. Bye-bye.
bye bye.